Great.
Live streaming is on.
Thank you, Zeb. Actually, there is a problem with my computer. No?
There's an echo here.
1, 2. Yeah, it's better. Thank you very much, Zeb, for the whole day to be with us from Dryfta. The platform is pretty stable all through, so it is much better than yesterday, and warmest acknowledgement as well. So we will come to session 6 on systems microbiology integration. It's the last session of the day. And Nina will mainly chair this session, and I will coach her and stay in support. So please, Nina, take the lead.
Thank you, David. So we will start with our keynote speaker of our last session. So Martin Duholm from Aalborg University will tell us a little bit about the MIDAS4 database. So it's a pre-recording, right? Yeah.
My name is Morten Duholm, and I am an associate professor at the Center for Microbial Communities at Aalborg University. I'm honored to be here today to give you.
Can we check that the YouTube link is available as well? It's there, it works, thank you. Sorry.
Good afternoon. My name is Morten Duholm, and I am a professor at the Center for Microbial Communities at Aalborg University. I'm honored to be here today to give you this keynote about the work that we're doing in our research group in relation to the MIDAS Global Project. I really hope that you'll enjoy this talk. I guess that we all know that microbes are the working horses in wastewater treatment systems. They feed on the nutrients in the wastewater, converting it to microbial biomass, thereby producing a cleaned effluent. And the microbial biomass produced can be used for the production of biogas and fertilizers via anaerobic digestion. It's therefore relevant to understand how these microbial ecosystem functions. And what we know already is that if we go into the biomass, we know that it's composed of thousands and probably even millions of different species. So how to deal with all of this diversity? So what would a biologist do? So a biologist would try to classify all these species, group them, and determine what are their functions. And all of this information could be turned into a field guide. In this case here, the field guide of microbes in activated sludge or in wastewater treatment systems. What should a field guide contain? First of all, it's important we have some kind of reference for the species, and that would be the names or the taxonomy. Next, we want to learn something about the phylogeny. How are the different species evolutionarily related, and with whom do they share functional traits? We also want to learn something about the habitats. Do they only occur in wastewater treatment systems, or are they also found in other habitats? What are their roles in the wastewater treatment? Are they involved in the degradation of certain compounds, or are they involved in the formation of tight sludge flux, for example? Also, to have a good field guide, we need to have images. We want to see how these species are looking. We also want to learn something about the interactions. Do they share nutrients, for example? How are they regulated? Do they require certain environmental conditions, certain pH levels, certain salt concentrations, and so on? And there could be a lot of other things that are important for these species. The idea is, if we answer these questions, we can actually pave the way for microbial informed management of wastewater treatment plants. We can improve the wastewater treatment plants we already have and even develop new treatment technologies. But to make this field guide, the first step is to get the species names or the taxonomies. How can we identify the microbes? Well, it would be easy if we could just culture them and study them one by one. Unfortunately, that is not possible with the microbes in the wastewater treatment plants. It's very few that we can actually culture. So we need to go to DNA-based method instead. And the idea by DNA-based method is simply that we want to collect genome information from these different organisms. Unfortunately, getting these complete genomes are very hard. So instead we use marker genes. And one really nice marker gene is the 16S RNA gene. And the good thing about this gene is it acts as a molecular fingerprint for different bacteria. You can actually tell bacteria apart by this single gene. And the more related the bacteria are in terms of evolution, the more similar are these fingerprints. So when we are profiling a microbial community, we are actually not using this whole 16S RNA gene. Instead, we're using shorter amplicons. In the Global Spiders project, we've used the V1 to 3 region and the V4 region. And because these sequences contains error when we sequence them, we need to handle that in some kind of way. So there's 2 ways to do that. The first one would be to create original taxonomic units where we cluster similar sequences, usually sequences which are more than 97% similar, into these OTUs. An alternative would be to try to infer statistically the exact biological sequences. That's called amplicon sequence variants. And that's actually what we are doing in the MIDAS Global project, because it has a higher resolution in terms of taxonomy. I would like to go a little into details with how we obtain community profiles. So the thing that we do is we take a sample, we extract the DNA from this sample, and then we prepare it for sequencing, and then we do some bioinformatics. So the first part of the bioinformatics is we create these amplicon sequence variants, and we actually create a table based on these amplicon sequence variants, which shows us how abundant are different amplicon sequence variants in different samples in terms of relative abundance. This is called this ASV table, as you see here. The next thing we need to do is to classify the ASVs, figure out what are the taxonomy of the bacteria behind each ASVs. And to do that, we match the ASVs up against a reference database. That could be the Silver database, the IDP database, or Green Genes. And then you get a list of taxonomies for each of these ASVs. And then we combine that with the amplicon sequence variant table, you actually get the community structure for your sample. However, as you can see over on the taxonomy table here, I have highlighted some ASVs in red, and that's actually ASVs which are poorly classified in terms of taxonomy. And it's important that we can actually go to this genus or species level in order to link the ASV with some kind of function for the bacteria. And the reason that we can't do that is actually due to the databases. So the databases are incomplete. They could either have no matching sequences for the ASVs, or the sequences in the database might not have any taxonomy, and it actually gives the same kind of problem. So what we really need is better reference databases. This was actually the reason that we created the MIDAS databases. So in MIDAS 1 and 2, we tried to get a better taxonomy. So we used the Silver database as a reference database, and then we just curated the taxonomy for common genera found in wastewater treatment systems, and provided placeholder names for some of the genera which have not been named. However, this approach has some issues. So first of all, it's a very large database, so it's computationally demanding to work with. It requires a lot of resources to update. So one person requires a month of work to actually update from one version of Silver to the next one. And when we do this classification, it's very subjective. So the person curating the database has to make a lot of decisions. Also, the taxonomy is not comprehensive, so we only have this genus level resolution for the common genera and wastewater treatment systems that the curator has actually curated. And there are no species level taxonomy, so we can't go beyond genus level classifications. And finally, because Silver is a universal reference database, it's not really suited for wastewater treatment systems. So there will be a lack of high quality references for many of the amplicon sequence variants we see in our ecosystem. As you probably know, DNA sequencing have really changed the last couple of years. So a couple of years ago, me and some of my colleagues developed a new method which allowed us to create millions of high quality full-length 16S and 18S RNA gene sequences. And what we did in one study was that we created more or less the same amount of sequences that there were in the Silver database. And this provides some new possibilities. We can actually now start to create a database where we have all the reference sequences for all the organisms within the ecosystem, for example, in wastewater treatment plants. And this method here have been further developed so it's more applicable for everyone. So it can now be also done using nanopore sequencing and PacBio sequencing. So now that we're able to obtain more or less all reference sequences from an ecosystem, we also needed to find a way to deal with the taxonomy. And to do this, I created AutoTax together with Casper, who's really good at actually taking my ideas and turning them into a program. And the idea behind AutoTax is that we take our full-length amplicon sequence variants, and then we match it up against the Silver database, find the closest relative, and obtain the taxonomy for these sequences, and that's what you see here in orange in the top. We also get the percent identity between our sequence and the ones in silver, and based on this percent identity, we can actually remove information from taxonomic levels which are not supported by this percent identity, and that's why you can actually see we remove some of the boxes here in orange. And because silver doesn't have the species level resolution, we also match our full-length American sequence variants up against a subset of the silver database, which only contains type strain. And these have a true species level classification. And that's what you can actually see here in yellow. And again, we remove the ones that we don't really trust based on the percent identity. And finally, we have some gaps in this taxonomy. To fill that up, we do de novo clustering of American sequence variants, similar to what you do when you cluster OTUs at different taxonomic levels. And that's based on percent identity again. So for example, if you go to the species level, we use 98.7% identity. And this creates this blue taxonomy here. And you can actually add that on top of the taxonomy and then get this merged taxonomy on the right, which goes from the kingdom level to the species level for all reference sequences. So if you want to learn more about this technique, you can actually go to our paper and read about it. Also, this technology here was what actually was used to create the -3 database, which is based on Danish wastewater treatment plants and anaerobic digesters. Although the -3 database works very well, even for global samples, it's still only based on data from Denmark, and there could be a lot of diversity which we are still missing. We therefore conducted the MIDAS global sampling campaign and obtained samples from wastewater treatment plants all over the world. So from more than 700 wastewater treatment plants, more than 400 cities, and more than 30 countries. And this is of course only possible due to a lot of good colleagues who helped us sample all of these wastewater treatment plants. And I would also like to give a special thanks to Vibeke, who coordinated this sampling, and to Simon, who processed the samples in the wet lab. When we look into the different plant types, we can see that the majority are using the activated sludge process, but we also have wastewater treatment plants with biofilters, MBBRs, MBRs, and granules. So we cover also the diversity within different types of wastewater treatment plants. If we look into the activated sludge plants, we can see that they are using different process types. So there are some plants which only remove carbon, some which also removes nitrogen, and then the ones with denitrification and the ones with biological phosphorus removal. In the -4 database that we generated, we had more than 5 million full-length 16S RNA sequences, and that was translated into more than 90,000 full-length amplicon sequence variants. In comparison, the -3 database was made up of nearly 1 million full-length 16S RNA sequences, but were only able to be resolved into 9,600 full-length amplicon sequence variants. And that's because we also improved the error rate of these sequences, and which allows us to recover more of these amplicon sequence variants. So now that we have this really nice database for bacteria and archaea in wastewater treatment systems, we wanted to investigate how much novelty were there in our database compared to the silver database. And the first thing to evaluate is sequence novelty. So are the sequences in themselves novel? And we use the threshold for defining species, genera, families, and so on. You can see here on the left. And what we can actually see is that we actually get a lot of diversity at the genus and species level, but not by all the family. Another way to evaluate novelty is to look into taxonomy novelty. So how many taxa do we provide placeholder name for, or de novo taxa, as is given here? And what we can actually see is that on all the different taxonomic levels, we actually have a high percentage of de novo taxa. And if you look at the genus level here, it's more than 86% of all the taxa which are actually provided with placeholder names based on the auto tax procedure. So if we don't have this de novo taxonomy, we're not really able to talk about these taxa at this level here. So to evaluate our database, we wanted to figure out whether we had more high density references for the amplicons that we actually see in these ecosystems compared to other commonly used reference databases. And to make an unbiased evaluation, we used data from unrelated wastewater treatment plants, which are part of this global diversity study that you can see here over to the right. So what we did was that we simply mapped the amplicon sequence variants to different databases that you see on the graph here. And then we calculated the percent of the ASVs which had high density hits. That's hits with more than 99% identity. And you can see here that the MIDAS-4 database outcompetes all the other databases, although they are much larger than the MIDAS database. And collaborators would, of course, ask the question whether it makes sense to have been part of this MIDAS project here. And I can say that if we use data from our study, we actually get a much higher percent of the A suites which have high density references. More than 95%, or around 95% of them have high density references. Having the reference sequences is only part of the story. We also needed to know whether we had a better taxonomy, whether we were able to classify more of the amplicon sequence variants. So we used the same data set again, and then we classify the ASVs at the genus level. And you can actually see here that we get a much better classification with the MIDAS-4 database, where we're actually able to classify around 75% of all the ASVs, compared to the Silver database, where we can only classify 30% at the genus level. And that's have a huge impact, of course, on how we can actually evaluate the data afterwards. A really interesting thing about the MIDAS database is that it also allows us to go to the species level. So you can actually see here with the MIDAS-4 database, we are able to classify more than 40% of all the ASVs at the species level. This is not possible with any of the other reference databases. And again, we can actually see whether our own data is better classified. And this is, of course, the case. So with our data set, we're able to provide genus level classifications to more than 90% of the ASVs that we have, and provide species level classifications to more than 70% of all the ASVs. And that's really nice. I guess many would like to know which amplicon primers should be used to profile wastewater treatment plants. So with our data, we can compare the V1-3 amplicons with the V4 amplicons. And the graph that you see here shows the mean relative abundance of different genera when measured using the V1-3 primers on the x-axis and the V4 primers on the y-axis. And what we see is that most of the genera are actually equally well detected using the two different primer sets. However, there are some which are better detected with the V4 primers, those are the ones in red, or with the V1-3 primers, those are the ones in blue. And it's approximately the same number of genera which are preferentially detected with one or the other primer. However, if we look more into detail with the ones which are more abundant with the 1-3 primers, we can actually see that some of the most abundant ones are completely absent when detected with the V4 primers, such as Acidovorax and Diphlorimonas. The same is not the case if we look into the ones which are more abundant with the V4 primers. They are still detected with the V1-3 primers, just in lower abundances. And based on that, I think we can say that the V1-3 primers are generally better suited for microbial profiling of wastewater treatment plants. And it's also some of the data that we are going to use in the next couple of slides. But there are, of course, taxa which are only targeted. You can actually figure out which one if you look into them supplementary data in our paper. So one of the really nice things that our data can be used to is to study the effect of geography and environmental factors on the community composition in wastewater treatment plants. And what you see here is a principal coordinate plot where different spots is different samples, and the distance that they are apart from each other says how different they are. And you can see here we have studied different process types which are in different colors. And what you can also see is that different process types actually cluster in different parts of this plot here, showing that they are unique. And you can also evaluate that statistically based on this Adonis analysis. So the p-value states whether it's significant, the clustering here, and it is below 0.001, which means that you'll probably not get these kind of clusters by random. The R-squared value here, which is 0.05, tells us something about how much of the variance in the total data set is explained by this process type or this factor, and it's a very small amount. And we can do a similar analysis for other factors such as industrial load, temperature range, continent, and climate zone. And we, in all cases, see clustering effects based on these environmental and geographical factors. And for all of them, we also can see that the variance explained by the individual factors are quite small. We also see that some of the factors actually seems to provide the same kind of patterns within the data, indicating that there could be some kind of association between the different factors. So the take home is here that the activated sludge microbiota is shaped by many different factors, and many of them are probably associated in some different ways. So a really cool thing about our data is we can start to identify process critical taxa. So one example are the core taxa, and we define 3 different types. We have the strict core, which are taxa that are more than 0.1% abundant in more than 80% of the wastewater treatment plants. That will usually be general degraders. Then we have the general core, which are abundant in more than 50% of the wastewater treatment plants. And here we start to see some of the functional guilts. And then we have the loose core, which are taxa which are abundant in more than 20% of the wastewater treatment plants. And that's more specialized bacteria, for example, bacteria which are involved in phosphorus accumulation. Besides these core taxa, we also define something we call conditional rare or abundant taxa. And we define them as taxa which are more than 1% abundant in just at least one wastewater treatment plant. And these bacteria are more specialized degraders. They could be blooming bacteria, foamoformers, and other bacteria which cause process disturbances, but are generally not there in very high abundances. So what does it look like when we try to identify core genera? So the plot here shows you the different genera on the x-axis in how many plants they were observed, and on the y-axis in how many plants they were more than 0.1% abundant. And what is really striking here to me is that most of the genera are rare and in very low abundance. So you can see that the color scale here is logarithmic, and that actually most of the genera are observed in very few plants and never really abundant. So the core genera actually makes up a very few numbers. And the same is the case when we look into these conditional rare and abundant taxa. It's actually very few of the taxa which occurs at more than 1% abundance. And the idea is that we can actually do the same kind of analysis here on the species level or on any taxonomic level. So if we take a look at the core and the CRAT genera, we can see that it makes out a small amount of the total diversity that we see. However, in terms of relative abundance, these groups make out the majority of the total community. More than 75% of the community is composed of these different classes. What is also interesting is that we actually have provided de novo names for many of these taxa. So for the general core, it's 20% of all the taxa which are provided de novo names based on order tags. And for the loose core, it's 50%. We can do a similar analysis on the species level. And again, we see that the number of species which are core or CRAT is very small compared to the total diversity. But in terms of relative abundance, they still account for nearly 50% of the community. And again, we see that many of these important taxa are actually only detectable based on their de novo species names or the ones we are provided using order tags. So it's really important that we consider these de novo taxa to get a comprehensive picture of the ecosystem. So with this global data set, we can start to interrogate the diversity within functional with the nitrifiers. And what you see here is the mean relative abundance of these nitrifiers in different countries. And what you see is that for the ammonia oxidizing bacteria, it's mainly Nitrosomonas which occurs globally. There's also Nitrospira, which can be a common mox bacteria, and then also, of course, an ammonia oxidizing bacteria. But Nitrospira is nearly never seen in this ecosystem, except for a few in the UK and in Canada. For the nitrite oxidizing bacteria, it's mainly Nitrospira worldwide, but we also have Nitrotoga especially in Europe. You can also ask whether there are any ammonia oxidizing archaea, and unfortunately, we can't see them using the -4 database yet. But we can use the V4 data that we have and then classify it based on the silver database. And this is what you get in that case. What you can actually see is that ammonia oxidizing archaea is more or less only present in Malaysia and the Philippines. So if you're anywhere else, you can disregard this class. So a really unique thing about the -4 database is that it allows us to get species level classification for a large fraction of our ambiguous sequence variants. So if we look into, for example, the nitrifiers here, we can see that Nitrosomonas has a huge diversity. So there's many species which occur globally, and they all seem to be quite important. And there's actually more than 60 species here. So you can see that there are some which can't be on this heat map here. Nitrospira we saw was really not abundant, but it's actually also just a single species which seems to be around the world. For Nitrospira, we see that it's mainly two different types which are abundant worldwide. So that's Nitrospira defluvi, which is a normal Nitrospira. Then we have a Nitrospira nitrosa, which can be a common mox bacteria, which also occur in many countries. For Nitrotoga, there's more or less only two different species which can be identified. So there's a huge difference in the ones we can see, and the really cool thing is that then it can now actually start to interrogate these different species. So what we obtained with the Midas Global project was actually an outline of the field guide. We provided names for the different organisms and showed where they occurred, but we needed to add more information about their functions. And we can do that using high quality metagenome assembled genomes. So we already extracted or recovered more than 1,000 high quality metagenome assembled genomes from activated sludge in Denmark using sequencing technologies. And the idea is that we actually want to take that further and obtain high quality mags for core and CRAD taxa globally. And then we want to predict the functional metabolic potential of these genomes. And we want to create fish probes so we can actually visualize the microbes in situ based on the 16S RNA genes, both the ones in the database that we already created, but also the ones coming from the mags. And then we want to go into situ and actually validate these functional predictions. And then finally store all of this information in the Midas field guide. So what are the take-home messages? I hope I convinced you that Midas 4 is a unifying comprehensive reference database and taxonomy for bacteria and archaea and wastewater treatment systems. That it provides this genus to species level resolution for the first time, and it can be used to compare results across studies, even though different amplicon primers have been used. We also showed that there are these approximately 800 genera and 1,500 species which are globally important. And if we learn about those, we can actually predict most of the things which are happening in the wastewater treatment systems. And functional information about these tags are available through the Midas field guide that you can access through this link here. And then I'd like to say that we're also working on Midas 5, which incorporates reference sequences from anaerobic digesters from around the world, and we expect to release it sometimes in the spring of 2022. So I'll conclude my talk by thanking you for listening, and I hope that you'll visit our Midas field guide and actually see what we are doing there, and especially the Midas Global page. And if you want to keep track of what we are doing in the research group, you can actually catch us on these different Twitter profiles. And finally, I'd like to say a big thanks to the Midas Global team and all the country collaborators who helped make this project possible, and also to Per Helke Nielsen for actually securing the funding for making this huge project possible.
Thanks a lot for your presentation, Martin. I think it's very clear that if a lot of reactions online. I think it's very clear that if we want to classify microorganisms in these treatment systems, maybe I should wait.
Let's wait 5 minutes.
I was saying that I think it's pretty clear that. Oh my God. Let's just continue. Yeah, if we want to classify microorganisms in these treatment systems at a deep taxonomic level, it's very clear that we need a detailed and specific database. So I think it's great all the work that you've already done on the 16S RNA gene sequences. Maybe before I jump to any questions that the audience might have, I think we're all very eager to get this database also for metagenome assembled genomes, especially considering the increasing amount of work being done on metagenomics. How many MACs do you think you will need to get a representative database that can be used around the world?
So probably a lot. So 10,000 is probably the least amount you need. So we could see that there are these 1,500 species which seem to be globally important. And of course, we can track them based on the 16S RNA genes. But there will be a lot of diversity within the species when you go to the genomes. So to have a comprehensive database, I would say probably 10,000 max at least. And there could, of course, be special organisms that we need to take care of also.
Yeah.
How do you come to this number?
So for the species, we based it on the amplicon data and try to figure out which one were common in wastewater treatment plants, and also including these ones which pops up once in a while. So most of the bacteria actually in wastewater treatment plants are below 0.1%. So of course they have an added effect to the overall process, but by themselves they have very little effect. So we want to get a picture of the system, I think it's fair to actually just go for the airborne ones in a start, and then you can actually explain probably 80% of what is happening in the wastewater treatment plants, and that might be well for most of what we want to do.
When could we expect such a database?
For the genomes,
Yes.
so I would probably say that it'll probably take 10 years. I think speakers always would like to say that it takes 2 years because that's what's technological possible. But in terms of funding and actually getting the data qualified and getting it out so it's usable for users, I'd say 10 years would probably be what is real. Of course, you can get it for many already. So for these 1,000 we have made, and there's all other groups who also made some high quality mags. So I think it's coming, but to get the comprehensive picture, you need more.
Great. There's also some questions online from Dr. Gagliano, Christina. Will Midas 5 also include already published data?
So what we do when we create these databases is we build on top of the databases that we already have. So we're only using our own data because we need really high quality of these 16S RNA sequences. So we create these amplicon sequence variants, and a lot of the data which is publicly available, for example, in Silva, does not have that quality. So we have a problem, for example, with chimeras in a lot of 16S RNA genes. And the method we use are not having these chimeras due to the quality control we do and the special way that we create them. So we only trust at the moment the ones that we already have. So for the Midas database or the field guide, we would really like that everybody contributes and add articles where there are information about the bacteria so we can get that incorporated into our description of the bacteria. Because that's not something we can do ourselves. So we can, of course, try to keep track of the most important ones and the ones we are working with at the moment. But there are so many bacteria that we need everybody to contribute. And we only include information from studies which have been published so that we can actually track the information and make sure it has been curated.
Yeah, I see. Yeah, maybe another question from Dr. Julia Vierheilig. Thanks for the great talk and for the important work on the Midas field guide and AutoTax. And then the question, did you also include industrial wastewater treatment plants in Midas, or are you planning to do?
Yeah, so there are a lot of industrial plants in the data set also. So we didn't, so there are some of the plants which are also in the analysis here, which were 100% industrial wastewater treatment plants. And we have, of course, a lot of special plants which are not in part of our analysis because they are more specialized or so different from what is in the wastewater treatment plants treating municipal wastewater that we can't use them for direct analysis. But they are we still have the sequences for many of these organisms. Of course, what we can see is that if we go and take a look at some of these specialized wastewater treatment plants, there might be some of them which are not that well covered by the database or by any database. So in theory, we can do an analysis and actually check whether they have good coverage before we do an analysis. And if they don't, we can actually take those samples and add sequences to them from later on. So that's also part of the idea that we can keep expanding this database when we find new exciting samples.
Yeah.
Can you explain in brief terms what is the difference between amplicon sequence variants and operational taxonomic units? I don't think that many people know the difference so far.
Yeah. So the main idea is that we want to get rid of some of the errors which are there when you do sequencing. So when we do sequencing, we introduce a lot of small errors, and we don't want to have them in a data set. So one way to do that is to group sequences together, find the one which are most important, and then see is there some sequences which are very similar to that one. So they could be sequences with a few errors, but it could also be other sequences which are just in lower abundances. And you cluster them together, and if you use this threshold of 97%, you get something which is sort of similar to what you could expect for a species. So that you simply group things together which you would expect are behaving more or less the same. But of course, you lose some resolution when you do this clustering. So amplicon sequence variants is another way to handle these errors. So there you try to identify, again, the ones which are most important, and then you use statistic to figure out, is this a real sequence or is it sequencing artifact based on how common are this most common sequence compared to the ones which are having one difference between it and two difference between it and so on. So there you try to infer the correct biological sequences, so the ones which are actually present in nature. That provides the higher resolution, and it's needed if you want to go to the species level. You can't go species level with OTUs. You can get some classification at the species level, but it'll only be for one part of the OTU, the centroid of it, or the most abundant one.
Thanks.
Maybe a final question before we move on from Dr. Niti Teresa. Nice talk. Oh, hold on, just scroll down. Nice talk. In your opinion, in activated slush system, what proportion of the total microbial diversity have we identified with the MAX and the de novo approaches? I think this is something we all would like an estimate for.
Yeah, so based on MAX, I think for the Danish system, we are around 50% of the community covered by those. Then that's if you have these 1,000 MAX for just one country, and it'll probably not be the same for global studies. So there it's probably 30 or something in that range. And with this database with the 6NS, I think we are around 95% or something in that rate. But it comes down to the question of what should be the resolution. When do you consider that you actually have a good match or have a representative species in a MAG or in a 6NS on genes? So the higher resolution that you want to, the lower percentage of the community have you covered.
Yeah. Any questions from the audience, maybe? No? There's a final question. Maybe we can quickly ask that one. From Dr. Claudia. Thanks for the talk. I found MIDAS classification very useful to have a common taxonomy. But how can we use the data deposited from other works to compare?
So I think one of the great thing about this MIDAS database is that if you have data from an older study and that data has been published, so if raw sequencing data has been processed, you can always reanalyze it and reclassify it, and then you actually can compare it directly to what we have here. I think one of the major problem is when we have done amplicon studies in the past is that we make an article based on our data, and then the only reference point is the taxonomies we get by the sequences in that paper. Nobody's actually taking the raw data out and matching it against the data from other studies. So this here would be something that you can actually start to do with this MIDAS taxonomy. And I think a lot of old studies would have much more value if you try to reanalyze. So you can probably reanalyze the data from a lot of old studies and get some new exciting articles out of that.
Great. Thank you so much, Martin. We'll come back to you later during the discussion.
You're welcome.
So next we have Zihan Dai. I think he's presenting online from Northeastern University, and he will be telling us about the genomic resource for drinking water microbiomes.
Yeah.
Do we have Zihan?
Yes. I'm trying to sharing the screen, but it said my screen sharing failed.
Oh no.
So this means that you have to go outside the platform and rejoin the platform. And then when you rejoin the platform, you have to look on the top left. There are some buttons to enable video, sharing, and audio. Yeah. If you can do that in 1 minute, it would be great.
Yeah.
Because we don't have pre-recording from you, I don't.
Sorry about that.
No, but just try to rejoin and it should be okay. And Eric, there is a problem with the showing the screen. So you see that the Senate cell appears twice in a big and small screen, and there should be the presenters appearing on the slide set afterward. Zihan, are you back?
Yeah, I'm here.
Yeah.
You hear
Can
me?
you try to share your screen again?
Okay.
You have an error message or not?
It's just saying my screen sharing failed, and so the camera and the camera and the microphone are on.
So
I don't know why the
Camera
screen
is
sharing.
Sorry.
your camera is not on. So we hear you, but
Yeah, I mean I already gave the permission for my camera and my microphone. It's just my.
Now we can see you.
no.
Yeah.
Can you try to share your slides?
Yeah,
again?
so the thing is, when I trying to sharing my screen, it says the screen sharing failed.
Okay. So we have your slides.
Yeah.
Yes or no?
Yeah, I send it while the email.
Yeah, exactly. So we will come back to your presentation probably at the end of this session, so we can upload your slides on the computer.
Cool. Sorry about that.
Yeah.
Okay, so next we have the presentation of Yuba Wang from Northwestern University about the integrated omic analysis revealed differential gene expression and potential for cooperation between denitrifying polyphosphate and glycogen accumulating organisms.
Eric, there is still a problem with the screen. We don't see the slides. We don't see anything else than the Senate cell.
I think I have submitted the pre-recorded video.
It's good. We have Yubo Wang's presentation. It was good. It disappeared again. Yeah, it's good now. Thank you.
Always enjoy the series of talks in Miwei, and now I'm going to take the opportunity to introduce a bit of my work, which I hope may contribute to our conversations on the underlying mechanisms of unusually high nitrous oxide generation in denitrifying EBPR process. In the next 10 minutes, I will go through the research background, research gaps, research objectives, and key findings of this study. To make sure we are on the same page, I will start with a very brief introduction on polyphosphate accumulating organisms, PALs. PALs are critical microbe in phosphorus removal and recovery from wastewater. They could release phosphorus during the anaerobic phase and could reuptake phosphorus during the aerobic or anoxic phase. A subset of PALs, denitrifying PALs or DPALs, are also capable of denitrification using either nitrite or nitrate as electron acceptor. And in this study, we focus specifically on DPAL over nitrite, because by coupling with an upstream shortcut nitration, this bell process could achieve simultaneous nitrogen and phosphorus removal at a lower oxygen demand, lower COD demand, and also less biomass production. The lab-scale denitrifying BPR reactor was run over a course of 3 years with synthetic wastewater and a feast-famine regime by alternating CLD and nitrite feed streams. DPAL activity in the system was evidenced with phosphorus release during the anaerobic phase and phosphorus reuptake during the anoxic phase. During the anoxic phase, along with the reduction of nitrite is the accumulation of nitrous oxide. A range of 60 to 80% of influent nitrogen was converted to nitrous oxide. This ratio is unusually high comparing with less than 2% of influent nitrogen would be converted to nitrous oxide in typical nitrification denitrification neutron removal valve process. This high conversion to nitrous oxide is promising if the recovery of nitrous oxide is a goal, like in CANDO project to recover nitrous oxide as co-oxidant. However, if nitrous oxide is not recovered, if they are just released to the atmosphere, that would be environmentally detrimental because nitrous oxide is a potent greenhouse gas. In wastewater treatment plants, at least currently, a minimum conversion to nitrous oxide is expected. So, to be able to manipulate nitrous oxide generation to either maximize its generation or to minimize its generation, it's essential for us to understand why. What is the mechanism underlying this unusually high nitrous oxide generation? Genome-centric metagenome and metatranscriptome approach was employed in this study to uncover the underlying microbial processes. As a background information, granule formation was observed in the reactor during the long-term operation. The 3 metagenome datasets covered the bulk biomass, the granule biomass, and also the suspended biomass. Well, according to within cycle carbon, nitrogen, and phosphorus profiles, and also a qPCR test on transcription activity of the core denitrification genes in the system, sludge collected during the anaerobic phase, anoxic phase, and the aerobic polishing phase were subjected to metatranscriptome sequencing. In total, 32 high-quality MAGs were obtained, among which 3 co-occurring Acinetobacter population, 1 Campylobacter GAO population, 1 Chloroflax sota population, and 1 Proteobacteria population accounted for above 90% of the overall transcription activity. I would like to highlight here the most active population in the community, which is a PAL from a novel Acinetobacter clade. We name this clade as clade 1F, and as the one most active population in the community, we are curious about how the genotype novelty of this Acinetobacter population correlates to the unique phenotype features characterized. For example, the efficient phosphorus uptake over nitrite and also the unusual high nitrous oxide generation. We start our analysis with a survey on denitrification genes. There are 4 core enzymatic steps involved in complete denitrification, and one hypothesis for nitrous oxide accumulation is that denitrification metabolism may stop at nitrous oxide with a lack of North gene. Among the 6 most active populations in the community, the 2 Acinetobacter population of clade 1F and 1A have complete denitrification gene set, while Campylobacter GAO1 and Proteobacteria PR6 are putative nitrous oxide producers with a lack of North gene. Another Acinetobacter population of clade 1C and Chloroflax sota C87 are putative nitrous oxide consumers in the system. With this, we are talking about genetic potential, not real activity. So, one step forward to link such genetic potential to real activity is the transcription activity of the corresponding denatrification genes. The transcription analysis revealed a potential segregation of denatrification metabolism between pals and flanking populations. To be specific, both nitrite reduction and nitrous oxide reduction were dominated by the Acinetobacter pal population, while for nitric oxide reduction, it was dominated by the flanking population, especially the 1 Campylobacter GAO population and the 1 Proteobacteria PR6 population. As we may recall, both GAO1 and PR6 lack the genetic potential to further convert nitrous oxide to nitrogen, and this means that the dominant nitric oxide reducer in the community lack the capacity to further convert the nitrous oxide generated, and this may contribute to the high nitrous oxide generation in the system. Another pattern observed that may correlate with nitrous oxide accumulation is the imbalanced transcription of NRS gene and NORS gene. Although both these two genes are dominantly expressed by Acinetobacter populations, in whichever pal population the NRS gene is of much higher transcription than the NORS gene. This pattern may be more obvious with this bar plot, and in one of the Acinetobacter population of clade 1C, there is even no observable transcription of the NORS gene. With this, I'm going to switch gears and briefly introduce the potential cooperation between pals and the flanking populations in carbon metabolism. The 3 co-occurring pals are dominant contributors of extracellular EPS, while the 2 flanking population PR6 and CH7 are putative scavengers of extracellular organic compounds, and PR6 may also play a supporting role in granule biofilm formation. So the Acinetobacter pals and Campylobacter gal and other flanking populations may actively cooperate in denatrification metabolism, and the pal populations may also provide to the flanking population their carbon source. And here are several take-home messages. First, stable phosphorus removal and recovery was achieved in a lab-scale denatrifying EBPR reactor over nitrite, and unusually high nitrous oxide generation was observed. Second, a novel Acinetobacter population of clade 1F was identified as the most active population in this unique community. Third, truncated denatrification pathway and segregation of the nitric oxide reduction into flanking populations that lack the capacity to further convert nitrous oxide generated, and also the imbalanced transcription of NR and NORS gene in pals may all play putative roles in driving nitrous oxide generation in this system. And fourth, flanking populations, especially Campylobacter GAO1 and Proteobacteria PR6, may play essential roles in the system by detoxifying nitric oxide. And with this, I'd like to thank Professor George Wells, Dr. Han Gao, and all other lab mates in Northwestern University. And thank you all for your time.
Thank you so much, Yuva, for your presentation. I already see someone standing here. Maybe we let the applause go. Well, we have a question from the audience. Sergio, whenever the sounds are done.
Hello? Yeah.
Can you hear me?
Yes, yes. Thank you for the presentation. And I have a question about one of your last slides that you went quite fast and I couldn't really see. When you, like, how did you determine that the PAUs were the main contributors to EPS production? Because you show like quite a big figure, but I didn't have time to look at it properly.
Oh, to for for this part of analysis, we did a survey on genes involved in EPS synthesis, and for the three co-occurring Acinetobacter pau population, they dominated. Actually, they contributed 90% of the gene transcripts involved in EPS synthesis, and also secretion. So that's the dominant kind of the key evidence we relied on to draw this conclusion.
Okay, but then I guess that the genes that you're studying then are only focused on the polysaccharide synthesis, or do you consider other polymers from the EPS?
A protein and also polysaccharide. We actually referred a paper, a previous paper that introduced in that paper they have a kind of a collection of genes involved in the EPS synthesis secretion pathways. So we mainly refer to that collection of database to do this part of work.
Okay, thank you.
Thank you.
Any more burning questions can come back later in the discussion. Thank you so much, Yubo.
Thank you.
We can now go back again to Zihan. So if Zihan can still not share the screen, then David will share his screen and he can pass the slides for you, if you are okay with that.
Are you there, Zian?
Can you see my screen now?
Now we can see
super,
it.
perfect.
Sorry about the previous text
Everything gets solved with time.
problem.
So floor is yours.
Perfect.
Okay, cool. So I will start now. Good afternoon, everyone. My name is Zihan Dai. I'm now a postdoc based at Research Center for Eco-Environmental Sciences, Chinese Academy of Sciences in Beijing, China. Today, I'm going to talk about one of my PhD projects, Genomic Resource for Drinking Water Microbiomes, a curated collection of genomes and metagenome assembled genomes from the drinking water microbiome. This project is supervised by Dr. Amit Pinto, currently working at Georgia Institute of Technology in Atlanta, USA. The development of high-throughput sequencing technology and the availability of a high-performance computing resource enable researchers to study the microbial community in environments based on genome-resolved analysis. Earlier approaches rely mostly on key genes inferred from the whole metagenomes, without considering the genomic context. The enormous advantages were shown in genome-resolved analysis as compared to gene-centric analysis, which is able to achieve higher resolution and accuracy, provide more comprehensive gene contents, and used as reference for future gene-centric analysis. To reconstruct the genomic resource for drinking water microbiome, 185 metagenomes collected from full-scale drinking water distribution systems were retrieved, including public available data and the data generated in our group. These metagenomes covered 7 regions worldwide, including Singapore, United States, South Africa, Netherlands, Puerto Rico, United Kingdom, and China. Genome-resolved analysis workflow was applied on the data, including raw-read preprocessing, single or co-assembly based on sampling location, and metagenome assembled genome MAG reconstruction. After recovering MAGs, quality score was calculated according to this equation for each MAG. 1,297 MAGs of quality score higher than 50 were kept after quality control, and following deduplication was performed based on ANI threshold of 99%, resulting in 944 deduplicated genomes. According to minimum information about a metagenome assembled genome standard, among these 944 deduplicated MAGs, 93 MAGs are of high quality, 371 MAGs are near complete, and 480 MAGs are of medium quality. We further clustered these 944 deduplicated genomes based on ANI threshold of 95%, which resulted in 860 species clusters in downstream analysis. To explore the phylogenetic distribution in drinking water microbiome, taxonomic classification was applied using GTDBTK. 4 species clusters were classified as archaea, while 856 species clusters were classified as bacteria. This plot here shows the percentage of bacterial species clusters on phylum level. So here we can see Proteobacteria is dominant in drinking water genome collection, which is consistent with previous research results in the field of drinking water microbiome. And it is followed by Planctomycetota. Moreover, prophages were identified in reconstructed species clusters using CheckV, Vibrant, and Verso2. These species clusters, for example, Spingomonas, are commonly occurring in drinking water environments, indicating highly prevalent phage-bacteria interaction in drinking water microbiome. In addition, we also applied rarefaction analysis on the data. So this bar plot shows the number of genomes included in each species clusters. Here we can see 666 species clusters out of 860 contains only one genome, which were regarded as singletons. So based on this, we constructed rarefaction curve based on all MAGs and MAGs without singletons. We can see here the rarefaction curve based on all MAGs keeps going up, which indicated that additional species in drinking water distribution system remains to be discovered. But they are probably rare members of the drinking water microbiome, because the rarefaction curve based on MAGs without singletons starts to hit the saturation point. To investigate the co-occurrence pattern of the species clusters, we applied network analysis based on RPKM. Out of 860 species clusters, only 164 showed significant co-occurrence with each other, and finally formed 3 large clusters. Furthermore, unsupervised clustering was performed on these 164 species clusters based on presence and absence of KEGG orthology. Species clusters were divided into 2 clusters, and here in this PCOA plot, each species clusters is colored based on taxonomic information. Cluster 2 only contains species from Paticibacteria and Dependentia. The microbes in these 2 phyla are host-associated microbe with reduced genomes, which indicated that the metabolic clustering is driven by the genomic content capacity in drinking water microbiome. To sum up, here are 3 take-home messages. First, Proteobacteria is prominently represented in drinking water genome collection, followed by Planctomycetota. Second, core drinking water microbiome is accompanied by site-specific transient populations. Last but not the least, metabolic clustering differentiates global occurring host-associated microbes with reduced genome from other bacteria. Finally, I'd like to acknowledge all my colleagues in Pinto Group and the co-authors from University of Michigan and the University of Chicago for their contributions to this project. If you are interested in this project, here are my contact details. Thank you for your listening, and I'd like to take any questions you may have.
Thank you so much, Zihan. Yesterday we saw that there's still a lot to find out in the microbiomes of drinking water systems, so I think this is a good step forward. Does anyone have any question? No? Yeah, okay.
So what functionalities are you interested in for your next research step then, to make the bridge to what Nina mentioned about the comment of yesterday, about what functionalities we aim to target in the drinking water microbiome?
So at this moment, we are trying to figure out what is the co-function in all the MACs we found of high detection frequency. So we would like to see the distribution of the functions and then figure out like which one of interest.
How will you validate these functions?
At this moment, I don't think so. Like we would do the validation in lab. So at this moment, it's just like we would like to present this genome collection for further reference, and also we hope it would be a starting point to beneficial for the drinking water microbiome community to generate more and more metagenome, assembled genome data.
Any hint to bridge to Aalborg to make a Midas for drinking water?
if it's possible.
Thank you.
There's also a question online from Mr. Jiangzi Wang. Hi, thanks for the great presentation. How are the data sets collected? Are they all in the same sequencing depth?
So the data set we collected, because we already see the number of metagenome collected from the drinking water microbiome is quite few compared to the other environments. So basically, we are just selected all the public available data from the drinking water distribution, a full-scale drinking water distribution system. So we don't really I don't think they are all in the same sequencing depths.
Okay, thanks. And a final very quick question from Dr. Rooskanter. Can you tell us what were some of the most commonly occurring MACs, like the single MAC that was found 18 times?
So at this moment, I think in my slides, let me try to figure out. So the one at this moment we didn't really look into on the MAG level. So basically, we are still interested about the taxonomy, the MAGs which are detected in like frequently in different samples. So we are interested in the MAGs of high detection frequency across like different metagenomes.
All right. Thank you so much, Zihan. We come back to you also later in the discussion. So now we move on to Julie Hardy from Veolia in France. She's going to present on the metatranscriptomic analysis in anaerobic digestion microbiome exposed to increasing ammonium concentrations. And this should be a pre-recording.
And just a question to Zeb, do we have the YouTube link somewhere in the chat? It is there. Yes, it is. Yeah, thanks. Eric, we have a problem of projection in the Senate side. Yes, it's solved.
Anaerobic digestion inhibitors affecting microbial community. Glad to be here. So, good morning, everyone. My name is Julie Hardy. I'm very glad to be here. Thank you to give me the opportunity to present my work PhD. So today I will talk about metatranscriptomics. So the most common anaerobic digestion inhibitors affecting microbial communities and methane production are VFA, dioxygen, and ammonium. So all of them impact the structure of the methanogenic community and reduce the performance. So to better understand how the complex anaerobic digestion microbiome respond to disturbance, I developed a bioinformatics pipeline dedicated to metatranscriptomics analysis. So first, I present my experimental setups and the aims. So I disrupted 2 reactors by increasing ammonium addition. And during all the experiments, I collect samples, and all of them I extract messenger RNA to apply bioinformatics analysis at different scales, analysis of metataxonomics and analysis of metatranscriptomics. So this analysis permits to improve the ecosystem functioning. So first, I have measured physico-chemical parameters. So I present here the concentration of ammonium during all the experiments. So first, we have the initial steady state, the second level, the high level of concentration of ammonium. Then we go back to the second level of ammonium, and then we go back to the final steady state. Then I have measured the residual COD concentration, which increased at high concentration of ammonium here. And then I have measured the methane production yield decrease with the increase of ammonium concentration. And then I have measured the VFA accumulation, and we can see here an accumulation of acetate at high concentration of ammonium. We can see also a little accumulation of propionate with no accumulation of butyrate. And then we can go back to the next level of concentration, and we can observe that an accumulation of acetate, and there is no accumulation of propionate and butyrate. So then I focus on the dynamics of the taxonomic composition. And here on the top, I present the concentration of ammonium during all the experiments. And each color represents each concentration of ammonium. And then if we can observe the present community, so the DNA based, we can observe that Bacteroidota was in majority here in blue, followed by the firmicute and spirochete. Here the firmicute in red, and in light purple the spirochete. And then if we can focus on the active community, so the RNA based, we can see that Allobacterota in light orange was more than 25% of the total active community. So then I take all the active community composition here, so correspond to Allobacterota, Euryarchaeota, and Thermoplasmatota, and I observe the metagenomic active community composition. So we can see that the majority was composed by Methanoculeus, Spirillum, and Methanoseta here in orange. And we can observe that in high concentration of ammonium here, we can observe less Methanoculeus, but a majority of Methanoseta in light orange. Then I focus only on active microbial community, and I apply correlation between operational parameters and microbial characteristics. So I constructed co-occurrence network with a correlation of Pearson. So we can observe three clusters. The first one was composed of the majority of Bacteroidia. The second one was composed with the majority of Clostridia. And the third one was composed of the majority of Archaemethanogen, correspond to methanomicrobia. So then I construct different modules and I correlated these modules with physico-chemical parameters, like here. So if we can observe the module 1, we can observe significant positively correlation with the propionate. And if we can focus on module 2, we can see a significant positively correlated with the methane production. And if we can observe the module 3 composed of the majority of archaemethanogen, we can observe methanomicrobia in these modules, and it's negatively, significantly negatively correlated with ammonium. So to conclude, the identification of microbial cluster linked to the methane production and favoring VFA accumulation. And we can see too the ammonium impacted negatively archaemethanogen. So if we can focus on the expression of genes involved in a specific pathway metabolic, here it was methane metabolism. I compare each condition here, so with the initial stage. So I compare the high concentration of ammonium here with the initial stage and the two sample linked to the final initial stage with the initial steady state. So then, after this comparison, I select a specific transcript with a log 2 fold change superior to 3 and a p-value inferior to 5 minus 23. So I represent here the cumulated log 2 fold change with the different comparison with the initial stage. So the first one was to the overexpression of transcript common to all the methanogenic pathway here. And we can observe that the submit of heterosyl disulfide reductase was majority and overexpressed at the end of the experiment here in purple. And the methyl coenzyme M reductase get the same profile. And then I focus on the enzyme linked to the hydrogenotrophic methanogenesis. So here was the format deshydrogenase was overexpressed at the end of the experiment. And these two enzymes, deshydrogenase and reductase, appears at the end of the experiment here. And if we can focus on the methanogenesis based on methylamine, we can see the presence of these two enzymes at the beginning of the experiment at the high concentration of ammonium. We can observe this here. So we can observe the methyl transferase, and at the end of the experiment, we can observe to the 3-methylamine co-methyl transferase. And if we can focus only on the methanogenesis based on methanol, we can see this enzyme at the end of the experiment only, and it was overexpressed. So at the end, to conclude, if we can we identify 4 specific pathways in the metabolism of methane. And if we can link the taxa with the information of functional, we can see that at the initial stage, we got the majority of methanochelys and a little methanospirillum. And then if at high concentration of ammonium, we get the methanogenesis based on methylamine linked to the high relative abundance of methanoceta and less methanoculeus and methanospirillum. And at the end of the experiment, we can see a majority of methanoculeus for both at the end of the experiment, and we can observe the link with the pathway metabolic. It was hydrogenotropic methanogenesis and methanogenesis based on the methadone. So to conclude, we can say that the pipeline permit to link the functional information with the taxonomy. And this pipeline can be applied with other study in different domains like environment, medicines, or other. So thank you for your attention. And if you have some question, you can ask.
Thank you, Julie. Are you here?
Do you hear me?
Yeah, great. Still applause is going on online. Does anyone have a question already? Okay, then I will ask mine. When you decrease the ammonia again in the reactor, did you see if the microbial community and the physiology as well recovered, or at least started to recover?
Yes, it just started to recover. Not a big recover, but a small recover we see at the end of the experiment. But there is a COVID, so we have to stop all of the experiments. So I don't have the time to show all the recover.
Yeah, but probably the recovery was much slower than the.
Yeah. Of course.
Therefore, we need autonomous sampling and analysis on reactors.
Yeah.
I have a question. So this morning we saw a talk of Nadi about metaproteomics and with labeling, and here you use metatranscriptome analysis.
Yes.
When should we use what? So why should we use transcriptomic in your case and metaproteomic in the case of Nadi? Maybe it's a question for the general discussion, but do you have any hint on why you selected metatranscriptomics and not metaproteomics or metabolomics?
Yes, there is a lot of scale to study the microbiome, but I focused on metatranscriptomic because it was a screenshot of all of the microbiome, complex microbiome. So I just want to see how they I don't know them. They move or with the comportment of this microbiome complex with the impact of the microbiome, the ammonium. Sorry. So I just want a little screenshot of this complex microbiome with the impact of the ammonium.
The question is it that you are more comfortable to work with RNA than to work with proteins or metabolites, or is it that you want to see something fast, and probably something fast change is more fast at gene level than at protein level?
Yeah.
So certainly some different types of kinetics that we can target with metatranscriptomics.
Yeah, because there is some RNAs they didn't. Some RNA are not reduced to protein, so we just want to see how the microbiome react to this ammonium. So we just want to focus on this RNA because it was more important for the study here. But we can add like after this we can add metaproteomics. It's more informative.
I don't say that, but what I want to say is that in metabolic regulation there are different steps of metabolic regulation. So that something happens between genes, DNA and RNA. Something happens between RNA and protein. Something happens after the protein synthesis to deactivate the proteins, and something happens with the metabolic fluxes. So when we select one method, we only get a snapshot on what happens with the
Yes.
translation between the gene and the RNA. If we would do something after the protein, we only see the regulation that happens after the protein.
Yes, of course.
There should be a clear research question with that. Your research question was clear, but when we select a method, it's important to keep in mind. I don't go against your study. I just mentioned to.
No, yes, because we want to know how they try to produce protein to with this perturbation only in this study, in my study. Yeah.
Maybe one short question.
Yeah.
How Veolia would use this data? So is it purely for research, or Veolia is interested to get something from the process itself out of these types of data sets?
Yes, now it's purely research, but maybe it was applied for a pilot or something like this. Yeah.
Thank you.
You're welcome.
Thank you so much, Julie. We will continue. So now Anastasia Kostrycia from the University of Glasgow will present on the DNA-SIP metagenomics to recover active ammonia and nitrite oxidizers.
My name is Anastasia Kostrycia, and today I would like to present you one of my postdoc projects done at University of Glasgow on DNA-SIP metagenomics to recover active ammonia and nitrite oxidizers. Anthropogenic nitrogen originated from agriculture or urban runoff can often lead to the altered or lost biodiversity and general habitat destruction of the coastal ecosystem. Microbial processes within coastal ecosystem can help to mediate the nutrient load entering coastal waters via various steps of nitrogen cycle. Nitrification, where we oxidize ammonia to nitrite and nitrate with help of a various group of microorganisms. First step is usually done by ammonia oxidizing bacteria or ammonia oxidizing archaea, while second one is done by nitrite oxidizing bacteria, is responsible for such transformation with help of these genes. In this work, we study the effect of salinity as one of the main drivers on the IO, ammonia oxidizing microorganism community in the coastal area of West Island, and we sample 3 sites with various salinities. In general, there is inconsistency of literature data who is dominating in which salinity, while AUA are quite hard to culture, and in general, active nitrifiers have quite a low relative abundance. Therefore, the aim of this work is to recover active microorganisms along the salinity gradient with a targeted metagenomic approach. So we use DNA-SIP for our incubations of 3 different sediments with various salinities and amended with ammonia. So we fed C13 label substrate to our for our batch, and in order to label the actively growing nitrifiers, we also performed 2 types of control for false positive exclusion. We then extract DNA, where afterwards we were able to separate C12 and C13 fraction with density gradient, and then they were perform microbial analysis such as qPCR of all fractions of AMO-A genes and both applicon and the metagenomic sequencing. So the first question we were interested to answer is nitrification occurring on all of the sites? And we saw that ammonia was consumed in each of the sites while nitrate was produced. So yes, nitrification occurred. We wanted to see if AUA and AUB were growing over time in each of the incubations, and we saw that gene concentration of AUA, AMO-A gene, was higher in site first with a lower salinity, while AUB, AMO-A gene were higher at the K1, AK2 sites. So here you see the data for K1 site as an example, where relative abundance of AMO-A genes measured for top AUA and bottom AUB, and for each density gradient fraction recovered from the centrifugation of DNA from either C13 incubation, which are in red line, or C12 incubation control, which are blue line. The red highlighted area indicate gradient fractions that were pulled for the subsequent metagenomic sequencing of heavy fractions. We also pulled together light fraction in a blue highlight area for control and a green circle for centrifugation control. So the same was done for each of the sites and in triplicates. In total, we did 26 samples. So I have prepared myself metagenomic libraries with help of Kappa Hyperplus kits. This is PCR-free, low-bias enzymatic fragmentation library preparation kit. You see here the average library size I obtained. Then we send it for sequencing in Illumina Nova 66250 PE platform, where we obtain about 31 million reads per sample, and then we used Atlas workflow to process our metagenomic data. So we used our amplicon sequence data for this experiment to inform our metagenomic targets, where we were able to see 9 AUB and 12 AUA amplicon sequence variant, which were at the same time underrepresented, a high active and successfully C13 label groups of microorganisms in each of the sites. And we wanted to see how far we can recover those in metagenomic data set. So after successfully processing data with our Atlas pipeline for metagenomic data set, we went for contig-based prediction in order to pull out ASV sequencing from our amplicon data set. So this was done by running BLAST against reference nitrification database for AUB, AUA, and NOB based on sample predicted genes, and we generated BLAST result here in a table. Then we took contigs with MOA gene sequencing out and aligned them against our ASV's database. We download the sequencing from our reference amplicon database, extract the same region of MO found in contigs in metagenomic database, and update our reference database with those. Then we built a phylogenetic tree for AUB and AUA, and with those you see our ASVs from amplicon study and other sequences are from contigs. Therefore, we see quite some clustering, and we would be able to rebuild some ASVs. And this is what we are currently working on at the moment. So we can use these findings for engineering systems as well. There are a lot of type of high saline industrial wastewater rich in ammonia, which can result in saline municipal wastewater. And traditional physicochemical methods are quite costly and energy demanded. Therefore, we could use biological treatment as an alternative and cost-efficient methods. However, biology is very often sensitive to salinity, and therefore we need a better understanding of the metabolism of different nitrifiers under varying salinities in order to optimize such a treatment. Thank you for your attention, and for further questions or collaborations, please contact me through email or Twitter, either about metagenomics, nitrification, salinities, saline wastewater treatment, etc. And I would like to thank Professor Cindy Smith and Dr. Umer Jaz, with whom this research has been done at University of Glasgow.
Thank you so much for your presentation. Anastasia, are you there? If someone already has questions. Not here? Not in person? Yep. Online also not? All right. Anastasia, are you there?
Yes. Do you hear me?
Great. We hear you. I don't see any questions. I have one, actually. How do these microbial communities, or basically the nitrifying community in these saline systems, compare to non-saline systems, for example, in wastewater treatment? Do you have any idea?
Yes, in some sense, it's one of the main factor which is determine just competition between different groups. So it's more that typically we see, as spoke today in typical wastewater treatment plants, some AUBs and COMAMOCs. What was speaking today with a key speaker, first one in the session. However, here in sediments, we can also see in quite some AOA, ammonia oxidized and archaea. So nature is always a wee bit richer than the system, I think, human-made. So yeah, it's much more varying and much more diverse what we can see in sediments.
Okay. Do you think this knowledge could maybe also be applied in wastewater treatments? If, for example, there are periods where nitrification fails because you have high salinity peaks for some reason coming in through the influent?
Yes, so generally trend is with climate change that the groundwater levels depleting and its intrusion of seawater into the continental area. So it might result in increasing salinity in groundwater, which already occurred in US nowadays. So it's going to be global trend that wastewater treatment plants going to have varying and increasing salinity over time. So I think this would be very perspective for future studies.
Great. Thank you so much. I don't know if anyone else has any question. Otherwise, we can
I do have a hand up. Can you hear me okay?
Yeah.
Yeah? Okay, cool. Thank you. A very nice talk. I'm Ryan Ziels at University of British Columbia. I just wanted to commend you on conducting DNA SIP metagenomics. I think it's a very exciting method that we don't see too much in these kind of conferences. My question is kind of related actually to the data analysis. Because it's sort of a new method, there's not really a standardized approach for how do you identify what organisms have incorporated the label. And so can you elaborate just quickly on how you identified incorporators using your pooled metagenomes?
So yeah, thank you. I know quite some of your studies on AD and stable isotope labeling. So happy to see you here as well. So we did perform this very carefully designed experiment with a lot of controls first of all, and then we did some amplicon sequencing and qPCR data. So qPCR help us a lot to quantify the dominance of different groups. And actually, in this type of study, I think there was some problem with the sharing screen. So when we did qPCR quantification for AUB and AUA, we did saw that there were distinctive peak between C13 qPCR data and C12. So, and then when we fractionate, we were able to pull the fractions which were well separated from controls with unlabeled experiment. Therefore, I think to having enough controls, having not only metagenomic data set, but also quantification of AMO-A gene, as well as our metagenomic database were informed by amplicon functional gene sequencing, it helped us all to connect all the dots and connect, make sure that the groups we gonna study are the ones which are active and doing this metabolism.
All right. Yeah, that makes a lot of sense. Thank you. Yeah, thank you.
May I ask one more question on this talk? Okay,
Very
so
quick.
For the DNA SIP kind of label of nitrifiers, where you choose the element of carbon, not nitrogen, like you labeled carbon to 13 carbon, why not 14 nitrogen to 15 nitrogen?
Okay. Yeah, thank you for your question. I think I might forget to mention it in my presentation. It's a very important point. So nitrifiers are autotrophs. So their assimilating pathways include CO2. So that's why it's very easy to label carbon, and it's much easier handling to work with heavy label carbon. It's most used isotope, heavy isotope. You can still do nitrogen, but it's more tricky, and it just you always go for the simplest available option. And for nitrifiers, because they're autotroph, we just can go for carbon. But for other type of nitrogen pathways where they don't have similar autotrophic processes, we probably will have to use nitrogen cycle labeling, nitrogen element labeling.
Thank you.
Thank you so much. I think we will come back to the methods later on during the panel discussion.
Thank you.
Finally, today we have Sergio Tomás Martínez presenting in person from Delft University of Technology. And he will also be talking about isotope labeling, but in proteins, specifically for tracking turnover of extracellular polymers of Candidatus accumulibacter.
Okay. Okay, now. And hello, everyone. Did you hear? Hello, everyone, and thanks for coming. Just in case you were missing me in the moderator table today, I'm here now, but presenting my research on stable isotope labeling to track the EPS of PAUs. I hope everyone is familiar with PAUs, also after the previous presentation, because I'm not going to focus on that, but mainly in the EPS. So as we all know, bacteria mainly grow in the form of biofilms, either attached to surfaces or granules or flocks, and embedded in a matrix of extracellular polymers and quite complex, where we can find proteins, polysaccharides, or other polymers such as nucleic acids and lipids. And in my research, I studied these polymers in one of the most well PAUs, Candidatus accumulibacter, due to its importance in aerobic granular sludge and the EBPR system. And we normally see these polymers that the bacteria produce themselves as a product of the metabolism, but we could also wonder if these EPS are stable, or they could also be a possible substrate for other organisms present in our enrichment or in their own environment. So in order to do that, what we did is to study the turnover of these polymers, and what we mean by turnover is, and what we want to see is the rate at which they are recycled. So we would see them as if from a substrate we would produce them, and that then they would also maybe be used either by the same organism or by other as substrate. If we calculate these turnover rates and we compare of these polymers, then we can compare it, for example, to what we would consider the theoretical turnover of or SRT, but that would include also other, not only the growth rate, but other decay rates that are happening in our biomass. But also we can compare these extracellular polymers with the intracellular and see if there are differences happening outside or inside the cells. And also, as I said, like there are many different polymers in our EPS. We could compare, for example, the two main polymers that would be like proteins and polysaccharides. Although today I'm gonna only focus on proteins. So in order to do that, what we have to do is to find a way of and differentiate what is newly synthesized versus what it was already there. And for that, we use a stable isotope labeling, as it could be carbon-13, nitrogen-15, or deuterium. In my case, I focus on carbon-13, and what we would have is a feeding of carbon in our system, and slowly we would see an incorporation in our polymers. So with that, the approach that we followed is we had an EBPR system, lab scale, that once we reach a stable state, we switched to, well, it was only fed with acetate, so we switched to fully labeled acetate. So in that way, everything that would be produced now would incorporate this label. Then we took samples after each of the cycles during one SRT and extracted the extracellular polymers and analyzed them through mass spectrometry and see this incorporation of the 13C label over time, and in that way check the dynamics of that. So as I said, this is the starting point that we had. It's a 1 liter lab scale and EBPR system, and we were having like normal PAU activity when we see like in blue the rapid acetate uptake and followed with the phosphate release, and then in the aerobic phase the phosphate uptake. And also if we check our granules, we see in blue here the all the bacteria, and in red the PAO that together do this magenta that shows that there is quite some PAOs in our system. So on this reactor is where we started to feed the 13C and analyze during one SRT all the samples, like several samples, like one at the end of each of the cycles. As I said, I focus on the proteins in this presentation. When we analyze them through MS, we see two different type of main data that we get. One is the relative isotope abundance, that is that shift that we see in the MS profile, that indicates that it's incorporating a label. And the other one is the labeling ratio, that it's this like 13C over the total protein that we can quantify, that it increases over time. So if we focus in the RIA, we see that over time and more percentage of proteins were incorporating this label that was showing that indeed the experiment was working, and each of the time series we were able of seeing more label incorporation. If then we focus in the labeling and ratio, what we would expect, so generally, and this type of studies are done in pure cultures, but also here we have an example in activated sludge, we normally see this kind of trend. And if we would consider that nothing is happening in our reactor, we would find this curve due to the washout of the polymers and new synthesis. However, when we see our data, we see that they don't follow that trend at all. So we clearly see two main differences. One is the difference on the total label incorporation. So that clearly shows that something is happening in the reactor. There is not like just like leaving and new being synthesized, but there is some kind of recycling. And then another important thing that we see, it's this delay that took around 3 days to really start like the same trend as the other ones. We think that maybe the fact, like if you remember from one of the previous presentations, PAO have like two main carbon polymers, that is PHA and glycogen. So at the beginning that can dilute with 12 carbon, our 13 carbon that we are feeding in the reactor, and maybe that is the reason why we see this type of delay. But we clearly see that it's a sigmoidal, so we use this type of sigmoidal equation to describe our data, that we see 3 parameters, and A that represents the maximum labeling ratio that we would obtain, that we would have obtained it if the experiment would have lasted longer. Then we have a kinetic parameter represented by B, and lastly C that represents this delay that we were talking about. However, due to our extraction method, that it's a quite harsh method to ensure that we get all the structural compounds in our EPS, we also get a lot of intracellular compounds. So in this case, for proteins, we used sequence-based methods to differentiate between what is predicted to be intracellular and what is predicted to be secreted. And in that way, we could compare extracellular and intracellular turnover. So this is what we see in this figure, that we see that if we check the kinetic parameter B and the delay C, they are quite similar. The main difference is in the level of incorporation that they show. And what we would expect if extracellular polymers and proteins in this case are being like consumed by others and being recycled faster, we would expect a higher turnover. But in this case, we don't see that. We see the opposite, or well, they are pretty similar at the end. So that would show that extracellular proteins are at least as stable as the intracellular ones. So, and that doesn't go with the idea that we had at the beginning, at least for the proteins. So as take-home message, I want to show that, and indeed we can use these long-term and 13C feeding strategies to study the turnover of polymers in this type of enrichment, that we see in our data a delay of the incorporation of the 13C, probably due to the polymers and intracellular polymers. that in the proteins, the labeling ratio, it's higher than what we would expect and due to the SRT, and also that if we compare extracellular and intracellular, we don't see that much difference as expected, with would indicate that their stability is similar. And lastly, I would like to thank everyone that participated in this research, and that is my student Erwin Swolzman, and also the PIs from our department, Martin Pabst, and my supervisors, David, Jume, and Mark, and everyone for your attention.
Thank you so much, Sergio. Are there already any questions online or on site? Some? Still the reactions going?
Is this on? Not yet, right?
Yeah, I think you can speak.
Is this on now? Yes. Perfect. So thank you, Sergio, for your presentation. You end your presentation with a sort of tantalizing question, saying that you have the stability for proteins in the extracellular proteins, at least. But do you now, based on the results you have, also expect all the other components of the EPS to also have the same stability? Or would you expect something else?
And indeed, like for polysaccharides, and at least so we will study the sugars that we expect to be in the extracellular matrix. I would expect the turnover to be faster than in the case of proteins, and maybe because also they are easier to degrade, but also in the study that I showed from like the EPS of activated sludge, they don't differentiate between proteins and polysaccharides, but C compounds and N compounds. So they see a faster turnover of C compounds, that you could think that it would be more like in the sugar side, than N compounds or proteins. So I hope to have the data soon to see this, but I would expect that turnover will be at least a bit faster.
Okay, that would be very interesting to see. Thank you.
Thank you. Yeah, there you can go.
Yes, hello. Nice study. I was wondering, now you looked into the proteins, intracellular and extracellular. So I guess you have also tried to identify all those proteins, so you actually know what it is. So and there could be some enzymes for degradation of proteins, but there could also be some enzymes to produce polysaccharides. Could you see any? You gave it an average, I think, but maybe I would expect that you had differences. Maybe could you see any differences?
And indeed, we have a lot of proteins in our data. The ones that I showed intracellular and are mainly the ones involved in the PAO metabolism, so in the PHA synthesis in glycogen and also in the polyphosphate. But I still have to go like we have like many of them annotated, but I have to go like deeply on that to see if we see more differences in a certain type of protein or in the other.
All right, maybe one final quick question before we move on to the panel discussion. From Ms. Xiaoxia Liu, EPS is often associated with stresses. Do you plan to apply some disturbances?
And yeah, like in this one, we wanted to just like see the turnover under stable conditions to see if something like if under stable conditions, for example, in the reactor level, if it was being like changed more often than we would expect. Indeed, if we applied some stress or some changes in the reactor, we would see maybe more changes, but at least in my side, I'm not planning to do that. But yes, it could be something that we could have a look at.
All right. Thank you so much, Sergio. You can stay there. Yeah, we can now have maybe a little bit more of a general discussion. It would be nice if all the speakers could turn on their cameras and maybe also some people in the audience so that we can see everyone.
And to save a bit of time, we'll do the closing in the same session. So also if Shaya is here, hi Yume as well. Sveko is still here or not? Okay. Or do you want to participate to the discussion, Ferike? Yeah. I think we make a closing of this session and the conference of the day at the same time.
Let's see, more faces popping up online.
And Niti, if you are online as well, you can also share your video.
Yeah. Okay, I think we can begin. I think we have seen not only in this session, but also in previous sessions today and yesterday that a lot of studies rely on omics techniques. And I mean on different metabolic regulation levels, targeted or non-targeted. So I think I mean, yesterday we also spoke about standardizing metadata and how important that is. So I think we also need to look a little bit into this in regards to the omics techniques, because it's a very rapidly increasing field. And I think we run into the danger that there's such a variety of methods available that everyone uses different methods, and this will result in a lot of biases in results, and we will not really have a very uniform analysis. So I would like to ask the speakers, do you think that methods are something that we could standardize better, and should we try to do it? And maybe you have any idea how? And perhaps we could start with Zihan and Morten, mainly on the metagenomic side. I think right now, or at least I see it already, it's quite problematic in a sense that there are so many methods available and so many different people using different methods. Feel free to jump in, anyone, by the way.
So actually, one of our goal for this project is try to optimize, because there are quite a lot methods now for assembly, for annotation, for like each step in the whole data processing workflow. So our goal is trying to optimize the workflow actually for drinking water microbiome, and try to figure out a way to retrieve and archive the data after publication, like processing it to reconstruct the max and then archive it in the database. So then we would have a uniform workflow to process all the data and finally make them as the reference database specific for drinking water microbiome.
Yeah. Morten, you have any comment on that?
Yeah, but I think it might be too optimistic to try to enforce that everybody uses the same strategy or assemblies of these data, because things are moving so rapidly. So we need to more actually come to terms with that. We need some kind of quality standards to adhere to. So, for example, the idea that we want to have other closed genomes, which is something we can't really do today, but probably in a couple of years, or at least go for the high quality mags that, in our opinion, need to have these 16S RNA sequences so we can actually relate them to also amplicon data based on the 16S RNA genes. So we also need to discuss how actually to create a database of all these mags. So even though we have created a lot of mags, we don't really have a central database for those mags. So they're being deposited together with all the other mags in NCBI and these databases. So it would be really nice to actually make it like part of the MIDAS database for the wastewater treatment system, where you can take out only the mags which are coming from this ecosystem and actually where you can rely on them. But it's not something which is easy in any way.
Yeah, absolutely. And sorry.
In terms of infrastructure, so generally groups develop their pipelines on purpose, on their database on purpose for the project. So here there is a name to be broader with MIDAS. So how to make it sustainably long, this use of MIDAS? So what are the computational resources that are needed, the human resources that are needed? You mentioned that to maintain Silva, it's already a huge computational effort. What would be it for Midas in the future?
So I think one of the ideas was to make this auto take procedure so we don't have to spend a month to update the database. But of course, there'll be a lot of updating when it comes to providing the functional information about all these tags, especially because there's this problem right now that the taxa is changing rapidly. So we don't really have firm taxonomic classification of many microbes because we're still relying on the functional traits of them or their appearance. So we're starting to get genome level classifications of species and definitions based on that. And that makes things more coherent. So when we get there, we can, I think it's something that we need to do one time and then just add additional information on top of that. And that's not going to be as resource demanding as it is nowadays.
So it means we need also bioinformatics laborantine in our labs now. So it shifts also the profession of laborantines from wet lab to dry lab. And in the future, we should also educate much more people on bioinformatics, not only at universities, but maybe in the University of Applied Sciences. And when they do their vocational training in companies as well, that there is more bioinformatics as well.
I think it's better to actually have it as a side thing. So you actually need to have both. I think a problem right now is too many people focus only on the omics techniques and the bioinformatics because that's what was easy and where you could get the easy points. So now we need to go actually back into the lab and actually validate a lot of the things we can predict based on genomes and so on. So I think we need people who are educated on both sides to actually to bridge the two fields.
I think there's some people online that want to jump in. Go ahead. Not anymore?
I think I would like to add, in terms of metagenomics, I show data on short reads sequencing, and our purpose was to assemble metagenome. And short read metagenomic doesn't allow, it's covering quite a short fragments. So it has a good quality, but the fragments are quite narrow. So in general, what I saw currently in a trend that short-gun metagenomics focused on short reads combined with something like nanopore, where you actually cross-validate both databases. So you have a quality and you have a length. So this is kind of cross-validation, and I think it can be that we can also inform our metagenomics from quantification qPCR data or amplicon sequencing as well, or use any other targeted approach.
Yeah, that's then another point. So when to use targeted methods and when to use general methods, because of course targeted methods in a sense are simpler, maybe because they generate a little bit less data. So you look very specifically to what you want to get, but maybe you're missing a lot on the rest of your community, because in the end we in this field want to analyze these communities as a whole, or at least how specific organisms react under certain conditions, which often also depends on how other microorganisms react. So I don't know if Anastasia, you or maybe Sergio, also working with targeted methods, want to comment a little bit on this.
I definitely agree. It depends on hypothesis. Depends on your hypothesis you put in the beginning of the studies. So it's it's quite different approaches. But the one I present today was a targeted. That's why I a bit referred to this type of approach.
Is it possible to always generate a hypothesis with these highly descriptive studies with metaomics? So if you look at functionalities, probably, but if you make more descriptive approach, how can you formulate a hypothesis up front?
No one wants to take that.
I think the two types of studies are valid, but I don't think it's possible in any case to put a hypothesis up front. But indeed, you need a research question that would lead you to select the right method to answer this question. Yeah.
And there is at the same time problems that you actually want to have, you always want to have a good coverage, and you want to have, so it means that you have so much data, it takes so much time to process it. So yeah, you will have so much data, and what do you do with it? So I face this obstacle now that we have 300 gigabyte data, which require 1.5 terabyte cluster. and we are like, it's gonna take so much more time to process more with co-assembly or something. So I think it should be, I still think it's important to put hypothesis a priority, because we generate so much data nowadays. Where do we even store it? Like, we just create, it's a, you know, form of online garbage in some sense. Not to, like, not want to say it in a bad way, but like, we create so much data. How do we store all of this and handle in the future?
It brings to the question, how can we integrate designs of experiments into bioinformatics as well? We can do that for wet lab experiment to minimize the number of experiments that we want to do to get some information that is already useful to answer a question. And I think in modeling and in bioinformatics, biostatistics, we can do the same as well. We could use these fractional factorial designs also to design dry lab experiments, let's say.
Hi, hello. I can say something maybe about that.
Yes, go
Hello.
ahead, Claudia, and then
Okay.
Shaya from Sweco.
Okay, thank you. No, just about the discussion that I think it's very interesting. I think also accumulation of data could help to answer some big questions. That's why I think we have to work on trying to standardize works. Then we can be able to compare. Okay, sorry.
No, you can continue, Claudia. There was a background person speaking.
No, sorry, because here there's another people talking in my office. Okay, no, it's about this. Maybe we need to work more also in this big database of marks, ensemble genomes. We also need to have metadata to be able to compare and to have and to postulate big question and then compare everything. I don't know if you understand my thought, but I think we are having a lot of data, a lot of sequence data, but we need more tools to try to compare them.
Shaya, you wanted to say something? Yeah. So actually, it is good to have a goal as a starting point and a hypothesis to start, and start with the first method you came up with during your proposal, I would say. But it's also important to adjust that along the way. So have more checkpoints, for example. And is it still valid, my hypothesis? Do I need to adjust the method or not? Or do I even have to adjust the goal? So I think more checkpoints will help to structurize our findings and approve the method, and also downscale maybe the idea and be a step closer to the practical application of the scientific findings.
And from your engineering duty at Sweco, both Ferike and Shaya, so how would you see that we could build such procedures? So if we need to make procedures, it's in research we don't like too much procedures, because as soon as it's ISO, I would say accredited, we cannot change anything. So we like flexibility, but based on your engineering lifestyle in your profession, what hints could you give to us?
Yeah, so protocols is a must in our work field. Otherwise, it's not accepted by the environmental agency. Of course, results need to be precise and validated. So I think the important part is to focus on key indicators, key markers, because the key ones, in my experience, they take longer. They can withstand a little bit more. I would say it takes much more to change a key indicator. So that sounds to me as a more promising. So find the key ones and try to standardize those techniques. And of course, a little bit of flexibility is okay, but as long as you have something to hold on.
So this brings to the term that we often use, that we need maybe an essential list of microbial information that we need to define for different types of system, at least in our specific projects or common targets that we have between different groups. And we saw, for instance, with drinking water field, it's not yet very easy to know what is this essential list of functionalities that need to be analyzed. But we need to work more on this essential minimal list that can help maybe to develop markers. So what is your thought, Ferike, as well on this?
Well, yeah, I think it's good to look at what methods are working and what methods are not. And I think also, like, now there are a lot of methods, but I think indeed what Shaya says is that you have to find a focus on a key point, because like the implementation is done by a lot of people who don't have an understanding of the microbiology behind it. So for implementation, it is important that like a person from the local government can also work with the standardization. So I think it's important to also keep that in mind.
Means we need to train our authorities to these techniques.
Yeah, maybe that's a good idea, but I'm not sure if I'm not sure if that's possible. But
Yeah, I think there are, instead of specialist conferences the one that we have here, there are some more general conferences also at IWA, at national level, at regional level. We had Aleida on Monday from Orvion, and she mentioned in the Netherlands there is the Federation of the Water Boards, and there they have also some joint meetings, and progressively we need to bring the results also to the authorities and the practitioners and to speak hand in hand with them. I think it's also well done in Aalborg, where all the research that you do is directly linked to the practitioners. Maybe Per, you have also an add-on on this. So what is the strategy to nicely bridge with practice and authorities? Maybe Mark also has an idea.
Yes, we have over the years made this developed this Midas concept. That's where we have a very nice collaboration to most of the Danish plants. I think more than 25 plants, and they have been really interested in knowing something about the community. So they have paid for now for 15 years to get new knowledge from us, and we have developed the new methods alongside. That has been an interaction where we have often meetings. We tell what do we know, what have we learned about their plants, and they have been very eager in learning something and provide samples and so on. So we have made a project we call Know Your Plant. So all of them sequence their plants once a week. They've done it for 5 years. They can go online and follow whatever is going on, and then they can try to correlate things to what they do at the plant. So this dialogue between me, all the people in our group, and then the plants, I think is really crucial. So and then they ask a lot of questions we have not thought about, and we can come up with some new stuff as they have not. They did not know anything about. Let's take example for immigration we heard about earlier today. We have worked with them now for 5 years because when we said, it seems the microticks may come from the infant, it's not the plant. They were, of course, very interested in knowing more about that. So this interaction between the questions they have and what we see has been crucial. Yes, I have one small addition about standardization. Morten, he mentioned that for Max, we should talk about the high quality. I have listened today, and yesterday we hear about a lot of Max, but definitions are very different. Some say 80%, some say something else, and whatever. But we do have standardization. It has been done by DBC or NCBI. Minimum standard for Max exists, so it's just to use it. 90% complete, 5% contamination, and 6S, 23S, and a number of others. So we should look in microbiology also to make sure that what we do is correct. We should not, in the engineering field, make our own rules. So we really had to stick to those standards as well. Yes, thanks.
Thank you. And Mark, from your perspective from the Netherlands, how you see that you can bring microbial concept and go further with these methods together with the water boards and the wastewater treatment plants? It's an ecosystem that is very active in the Netherlands because of the water boards that manage the water. Obviously, it's quite unique in the world, this situation. How do you see it different from Denmark? Is there a difference?
Well, there are of course there are differences. I think the Dutch water boards are currently, well, the Danish water boards are picking up, but have the last 5 years been much more busy trying to endorse resource recovery and process engineering developments rather than getting more knowledge of the plants. What essentially is what you do is microbial methods, and that has partly to do, of course, with the academic groups which are present with different attitudes, but also who's working in the water boards and the strong emphasis on resource recovery and aspects as limited sludge bulking is essentially hardly present anymore in the Netherlands. At least in the 90s, this was a heavy debated topic, and in practice that is more or less solved. There's sometimes a bit scum still in a digester, but there's hardly any and there's no problems with the operation itself. So it means that it's also difficult to come with blunt, wide-scale monitoring programs. So it has to be targeted or directed, like the project from Nina on N2O, then it was possible to include it in a smart way.
Thank you. And you mentioned bulking, so it bridged to early works of Eichelboom on the microscopy, and he was an awardee of MIWI, or the former MIWI, I think. So there was a huge work on microscopy that was done. To which extent do we make a difference with systems microbiology methods then?
Well, yeah, I'm probably the wrong person to ask, because I think Pavel Chudoba had already solved it somewhere in the, what was it, N60s. And since then, there's nothing new, and his methods work.
Thank you. So we're soon to close. Maybe it's time to wrap up the main messages rapidly. And Niti, are you online? And maybe you can give your perspective on what you learned today, and we can follow up tomorrow as well. Are you online, Niti? No, so I will give some messages, and if Niti will join tomorrow, we will do tomorrow. But for me, the main messages that I got today is that there is a whole power of methods beyond DNA. And there are plenty of informational molecules, DNA, RNA, proteins, metabolites that can be tracked. We can label, we can radio label, we can use other types of labeling with C13 and 14 analysis, etc. To track the functionalities of the population beyond the name. So often in publication, we see fingerprints with just phyla, and we think, what does it bring, this information, just at phylum level? So maybe it brings information, but at least it should be stated why. But we can squeeze the metagenomes if we have high quality mags, if we can combine mags with metatranscriptomics, metaproteomics, or other methods labeled, we can get much more information on these microorganisms and how it can link to process operation. We have seen with the terrific session on theoretical ecology and engineering theory that are not only deterministic phenomena, there are stochastic phenomena. We do think that we need probably a task group, a task force on theoretical ecology and water engineering to bring it further. So at the end of the session, I stay a bit on my hunger, and I'm still thinking, after 4 MIWI conferences, how can we go further with these theoretical ecology fundamentals? And probably we need to integrate that stochastic modeling together with traditional mathematical modeling of wastewater treatment plant or processes like that. The traditional model, like activated sludge model, will show the changes between the different types of equilibria, so the steady state, and then at steady state, at pseudo-steady state, there are some variations that can be characterized by stochastic modeling. So we need to have more modeling at MIWI, probably. It's clear in sets of talks that we received, there was not a lot of mathematical modeling approaches, but maybe we should bridge more with other specialist group, like the biofilm specialist group. The link is already very well made, but there is other groups on mathematical modeling, like Peter van Golechem and others, that we could maybe bridge further in the future to integrate our systems microbiology in modeling. As soon as we are able to integrate knowledge in models, we are able to rationalize the knowledge and to potentially integrate that with calibration, with pilot or full-scale system or bioremediation system, surface water, canals, big channels that we have seen in China, for instance. So it's not just about the phyla, it's about the functionalities and how can we make sense of them in a process. So thank you very much. I would like to warmly thank Nina and Jay for today's interaction. It went fantastically. Thank you so much, Nicole and Romy, as well, for the fantastic organization. Zeb from Dryfta Online, who helped us to have a pseudo steady state as well in terms of digital live streaming. Thank you very much, everybody, for your talks, your interaction. A warm applause again. For the on-site participants, Sam will give a small introduction for the dinner of tonight if you want to join us at the restaurant. So maybe you can come in front and explain us. And for online participants, we see each other tomorrow morning again for a free nice session more related to environmental health protection. See you tomorrow.